Okay, we're back. We're live. We're Think Tech Talks here at the SOEST Hour. Every Monday from 1 to 2, we do the SOEST Hour. And if you didn't know by now what SOEST stands for, I'm going to tell you. I'm going to tell them. School of Ocean and Earth Science and Technology at UH-Manoa. You know, and if you don't know by now, you should probably have a little tattoo made on the inside of your eyeball so you can remember, because it's an important school. It's a land-grant school, part of the Sea Grant college system at UH. And we have here from SOEST Christina Runyon, who is a graduate researcher and who you can relate to. You can relate to both of these guys, actually. Relatability is important, you know. Microbiology is not about microbes, it's about relationships.
Very good.
There you go.
That's really, I like that one. I'm going to use that one now.
Yeah, Christina, I'm not saying what the relationships are between. We'll talk about that. Christina Runyon, graduate researcher from UHM, Hawaii Institute for Marine Biology, and Blake Ushijima, graduate researcher also from UH-Manoa Department of Microbiology. So they're both working together. And they're working together on coral, and that's why we're calling this show Saving Our Coral, because, you know, coral has disease too. So just to get a handle on this, first, you know, before we get to the actual coral and the diseases and what we should be troubled about in that regard, why don't you guys introduce yourselves? Okay, Christina, go first. Tell them who you are.
I'm Christina Runyon. I am in the marine biology program at UH-Manoa. I am, this is my third year there, and I am working right now with coral disease on Kauai. I've had a lot of previous experience on Oahu, in American Samoa, and on Palmyra Atoll, looking at different diseases and different disease states that are there. And I've been very, very fortunate to be able to stay in the same lab since 2008.
Why?
Because I love corals. Well, it's not just corals. Actually, the first thing that when I came to Hawaii, what I wanted to work with is I wanted to work with marine mammals. But everybody wants to work with marine mammals. Everybody likes cute little dolphins, and competition's super fierce. You know, the one underdog is the backbone of all of our reef ecosystems around our islands. We are an island state, and that backbone is corals. So I started looking into being a coral biologist because it just was something that really intrigued me on how this little animal has such an immense impact on our ecology, our biology, the economic state, and it's very, very culturally important here in Hawaii.
Corals can be cute too. That's what I wrote down.
Absolutely. They're cute little flowers.
Yeah, it reminds me, you know, some of the aquaculture people, you know, they would never eat their own product. You know, you would never injure coral.
I
Never.
do my best to not injure coral. Yeah, that's another thing that we do is that we do report. Like when there's boat strikes, we'll go out and take images, send them to the proper authorities. We do our best to try to protect our reefs because
This is a permanent condition, actually, Christina. I mean, are you always going to feel this way about coral?
As long as they're around.
Ooh, good answer.
As long as they're here, I'm going to care about them. And not just them, also all the organisms that thrive within them, our fish, our invertebrates, anything, even our little bacteria, they're super important.
Could tell us just briefly what a coral is. Was it this big? This big?
Oh,
This big?
what? The individual animal?
Yeah.
It depends on the species.
Could be this big or this big.
You could have it at the teeny tiny tip of this little pen right here,
Yeah.
all the way to a plate of a fungia that's this big, that's one single polyp. But then when you have these guys where it's just that little thing right there, you can have like 10,000 in this little table area. And
What color are they usually
it depends on them.
when they're healthy?
When they're healthy, well, so the species of coral that we work with, which is Montipora capitata, normally it comes in a brown or an orange color.
Oh, pretty.
It's really pretty, yeah.
At least cute, anyway.
Oh, yeah, they're
and
very
by the way,
cute.
that's Montipora, Montipora capitata. God, you know, it sounds like something out of a religious ceremony or something. Montipora
All
capitata.
Latin, all Latin, 100%.
Okay, let's shift to you, Blake. Why are you here?
Well, I work on the other spectrum of this project. So Chris does a lot of the field work. She works a lot in, pretty much in the water, looking at the corals, looking at the different cases. I'm more of the lab rat, so I'm the
You
guy.
heard it here on Think Tech.
Yeah, so I'm actually in the laboratory. I study the actual pathogens or putative pathogens we think are involved in these diseases. So I'm the guy looking at the bacteria, and my main focus is on bacterial genetics and seeing what makes these little bugs tick.
So it's not just so much a matter of finding the bacteria,
Yep.
it's finding the genetics in the bacteria.
Yep.
You guys are really drilling down deep.
Yep, you want to know what really makes them tick, what makes them, how they kill, how they spread, just everything about this pathogen, so we can allows us to actually manage this disease in the future. Because if you don't know what's killing your corals, well, it's very hard to manage your corals.
Do you love corals like Christina
I
does?
do actually.
Do you hate the pathogens?
It's more of a love-hate relationship. So I hate what they're doing. As a microbiologist,
But they can be nice,
I
too.
do love bacteria. I do love studying bacteria. I don't appreciate what they're doing, but there's a little bit of admiration of how something so small can cause so much trouble
Yeah.
for the environment.
I think you could, well, can you beat them, you think?
Well, it's a war that's been being waged between humans and microbiologists and bacteria for centuries now. So I think we can fight back if we learn enough about them.
Fight back on
Yeah.
behalf of the coral.
Yes.
I love it.
Yes.
A war. So, I mean, you in it for the same kinds of reasons that Christina is in it? I mean, this is a lifelong commitment to coral?
Well, yeah.
Or is it a lifelong commitment to bacteria?
I would say both. I was born and raised in Hawaii, so of course there's that relationship with the ocean. You do appreciate, being able to just walk down to the beach. So I do feel that we should be taking care of our reefs. But then there also is the fascination with the microbial world, like how these little things can cause such great things happen in the big world.
Yeah.
So
Yeah.
it's a little. I do appreciate the corals, but it's a little more. I'm also I'm looking at the bacteria involved and.
Yeah. Well, I think, you understand, you both understand, but I think a lot of people in Hawaii who live, and spend a lot of time in the ocean don't really know too much about coral. They know about dead coral, because a lot of the coral around where they swim and surf and what have you is dead already, but they don't know how that happens, how the coral dies. And, I just want to get a handle on how bad this is. I mean, I can remember, for example, in Hanauma Bay, that was a big coral place. I'm sure it's not as big a coral place anymore.
There's still coral there.
Still coral there.
Yeah.
But there's also dead coral there.
Well, there's dead coral. Death is part of life.
Okay, fair enough.
Yeah, that is something that
But
we do need to understand that.
But you also get the feeling that, the coral that has died around these shores in Hawaii, which used to be so beautiful with the coral, those brilliant colors and all the divers and the snorkel whatnot and all that, that somehow, the population, somehow the intensity of the, of the populate community has, killed them somehow. But, so talk about that, will you? What has happened to our coral beds here in the last, say, since statehood? Okay.
Well, you can also relate it to the forests and the different, areas in Hawaii. It's been affected similarly with the expansion of, humans. We need to make rooms for malls and parking lots. So it's same thing that's happening to our reefs. So for an industry that's based on tourism, we do need to bring in the tourists to sustain this economy. But fortunately, you kind of have people stepping on corals, and to dispel some myths, they are living creatures. They are animals. So they're not some rocks that you can step on that they do feel it.
To step on them, they strike back. I've had that in my foot, yeah.
Oh yes,
They
there's
can.
the infamous coral.
Yeah.
But it's we do see some declines. We have people, we have networks like Eyes of the Reef. We have monitoring the state of the coral reefs. So I mean,
So how do you do that? What's the metric on it? How can you, if you go out to a coral reef on day one and you take a picture and it's alive and well, and then you go back on, 6 months later, and it's not so good. What's the metric?
Well, some of the things that we do is since we do evaluate different diseases in the field, and something that we got to remember is that all animals have the capability of contracting disease. It's the thing that concerns us is the levels that we're seeing, the disease.
This is new.
For
The levels of disease.
the levels of disease that we're seeing that are, it's not so much that it's new, it's just that it's increasing. We are seeing novel diseases pop up and such, but when you look at it on the basis of how much coral are we actually losing, it's really, really hard because we're not at every single reef. But what we do is when we do see a coral or a set of corals, or we see some sort of event where a lot of corals are becoming sick, we will go out and we will monitor them on a weekly basis, take photographs of them, and then bring those photographs in and use digital imagery analysis to track how much coral has died, or has the coral come back, or has the colony just completely collapsed.
How can you tell? I mean, if photographs or otherwise, how can you tell between a live coral reef and a coral reef that's not doing well? I mean, a dead coral reef, for example, can you see it in the
Oh,
photograph?
absolutely, you can see it. Because what you would remember that with the corals that we're dealing with, they're colonial animals, and they have all these little polyps across the whole entire colony. And if you look really, really close, and if you see a hole without a polyp, that's a dead coral. And you can differentiate the difference between, say, alive and dead. If you could put up the first image,
Yeah, okay.
then I can show you.
There's
So
one,
on
okay.
this image right here, this is tissue loss disease. This is the disease that I'm actually following in Kauai, and this is Blackband disease. That black line that you see there is a microbial mat that is creating an environment that is causing tissue loss.
The black line on the bottom, or sort of to the right?
See that what the pencil is pointed at?
Okay.
Okay, and then if you make around, there's a semicircular
Yes.
area. So that's the lesion. That's the lesion edge.
So what is a lesion? I mean, what's a lesion? Generally, a lesion.
For this image right here, the lesion is the area where there's tissue loss. Below that area, you'll see that there's a sort of brown color. That's the live coral.
Yeah.
Up above that line, that black line,
Yeah.
that is dead coral, which algae is settled in. So if you get your face straight up on top of that and look, you can see the polyps in the one instance, and you won't see the polyps
What's the
in the
scale
next.
on that? I mean, I see a hand over there with the pencil, so that whole thing looks like it's like 4 inches wide.
That whole thing is probably close to about 10 centimeters long, and it is progressive. It started out really small, and it has gotten to that point. The next image that I have up there, so this is another kind of a lesion, and this lesion right here is called a growth anomaly. So it's sort of like a tumor, coral tumor.
I'm feeling sad for them.
It is sort of sad. I mean, the disease states that we see, they are indicative of something not being right in the environment. If you move to the next image, we also have discoloration that occurs.
Now,
on corals.
what's in the middle there?
The middle there, so
that
that's-
looks healthy. Is that healthy?
Well, the coral itself has a parasitic infection.
Oh.
So that's what's causing that bright pink. That bright pink is not the natural color of the coral. That's an
Oh.
irritated part of
What's
the
on the
coral.
left? What's the
On
green?
the left, that's another coral that also, if you look closely, you can see there's little pink dots on it.
Yeah.
It has the infection, this trematodiasis, this little parasitic infection.
That's not bacteria now. That's something else.
No, this is something else. This is actually a little parasite that's made a little cyst inside the coral and turns bright pink, and it's calling for fish. Come eat me.
And the fish come around.
The fish come, peck at it. Certain species of fish, butterfly fish, comes, pecks at that little spot, and then that parasite goes into its next life cycle.
Is the coral injured by the pecking or the parasite or both?
Well, sometimes the parasites, if they don't get pecked at, they senesce and they absorb back into the tissue.
Which is not good either.
Well, I mean, it's not a really, really good, awesome situation for the coral, but it's not something that's going to outright kill the whole colony. But it's still something that we do look at just to see, if this is within the areas that we're surveying.
You took these pictures.
This picture right here is Greta Abbey. Greta Abbey is our primary investigator. She's one of the people who's in charge of us. So all three of the images I just showed are all from Dr. Greta Abbey. She oversees us.
I feel bad because, I mean, I know coral's important. Coral's important to the islands, not just tourism, but to the whole ecosystem. It's kind of like a canary in the coal mine, isn't it? If your coral's not good, your whole system is under some kind of duress.
Yeah, absolutely. That analogy's been used many times, and it does describe what's going on. It is our canary in the coal mine.
Wow, we didn't even talk about this.
I know, right? Gosh, this is awesome.
So that's Christina Runyon. She's a graduate researcher at UH-Manoa in the Institute for Marine Biology, which is part of SOWEST, and Blake Ushijima, graduate researcher in the same place. And we're talking about saving our coral. And here on Think Tech Talks in the SOWEST Hour, I'm Jay Fidel. We'll be right back after this short break. All right, we're back. We're live. I'm Jay Fidel here on Think Tech Talks, having a wonderful time on a Monday. It's like waking up, you know what I mean? It's like really getting alive, education, learning, and talking to these guys. It's thrilling because, you don't get to meet them that easy. And I have a great spot here because I get to meet them every week. Here on the SOWEST Hour, Saving Our Coral with Christina Runyon, graduate researcher at UH-Manoa, Hawaii Institute for Marine Biology, which is part of SOWEST, and Blake Ushijima, graduate researcher in the same place, talking about coral. And wow, this is, so central. People, they go to work, they're in their office or at school, they don't get out, they don't surf as much as they used to, they don't scuba, they don't snorkel as much as they used to. This is like the front line of our whole island. This is the place where everything meets. You guys are there, good for you. Anyway, so let's talk about more pictures.
Okay, so the next image that we're going to show you is, this is Blackband disease again that I'm working with on Kauai, and this is a different species.
But the line is the same.
The line is the same.
You use lights to get this picture, or is this natural light?
No, this is natural light.
And how far under the surface are we?
Oh, for this image, I took this image at about just 10 feet on snorkel. Just free diving.
Use a camera in a housing?
I use a camera in a housing.
What kind of camera
I
do you
have a Canon G12 that I use.
I'm trying to think. That's a pretty sophisticated camera. But you could buy it at Best Buy too, huh?
I don't know. I've never tried. I've only got them off the internet.
Okay.
I've never tried. But yeah, we use photography and
no flash involved here.
no, we don't use flash.
Why? Because that would hurt the coral.
No, because it doesn't do anything for our images. It doesn't really brighten them up or anything. You got to remember that inside of this animal here, we have some photosynthetic algae that could really just the flash, unless you're at depth, it really doesn't do you any good.
Yeah, okay.
The light's just going to bounce back at you.
All right, so you have the black line. It's the same process.
So you have the black line, same process, but this is a different species. So the species that I showed before was Montipora capitata.
Yeah.
This is Montipora patula. So
Catula, spell it?
Patula, P-A-T-U-L-A.
Patula.
Patula.
Like spatula, but without the S.
Yeah, exactly, like spatula without the S. And, you know, right now at this point, we've now seen this disease affecting not only the capitata and the patchlet, but we also have another, the blue rice coral, that it's also affecting in Kauai right now. And
where were you looking in Kauai? What part of Kauai were you
well, the
swimming?
first place we went up to was up on the North Shore, Nini Beach. And then from there, we just sort of spanned across the North Shore over the summer. And then this winter, we went and did the south-facing shores. You can go ahead and put the map up, and I can sort of
Oh,
talk
good
about
idea.
it.
Okay.
So, like I said, we
This
did.
is not a coral, and it's not a parasite or a bacteria. This is actually Kauai.
This is actually Kauai. That is correct.
Funny thing is, you could get confused about that.
I guess if you're not used to looking at satellite images, then yeah, you could get really confused, or hybrid satellite images. So what we've produced here is just a basic map of the sites that we've been to over the last year. So we've done a total of 38 sites, 25 up on the north and west-facing shores, and the remaining on the south, and a couple on the east-facing shores. All the red pins that you see on the map are areas that we found the disease at. So across the South Shore, we really haven't come across the disease. There's just one area that we've seen it on the east side, but we see a lot of it up on the
Are you
North Shore.
satisfied, though, that you have surveyed the island and you know where the places are that, you know, reveal the disease?
Well, I mean, we
Or do
would
you
like.
have more to go?
Well, we do have a lot more to go. I mean, for the next 2 years, I was fortunate enough to get some Sea Grant funding, and I'm going to be able to go. I'm going to be able to go 4 times a year, and we have semi-permanent sites where we are going to continue to revisit. We've established those on the North Shore, but we haven't established any on the South Shore. So this spring we're going back to establish that, and then from here on we're going back to monitor and to look at other environmental factors such as temperature, salinity, TSS. Look at the organic composition that's in the sediments, look at flow, anything that we can try to do to tease this out. And then we're going to use modeling and look at the spatial distribution, and then try to put out a product for management, just to sort of help them understand.
Management, like government, like DLNR?
Yeah, DLNR and DAR have been very instrumental as well. The trip, my last summer trip, and my trip that I just took this winter, they funded for us, which was awesome.
State government, that was good.
Yep, it was really
They
good.
should be concerned about the coral. It's a characteristic of the state, as it appears to people who visit us.
Yeah, absolutely.
So why did you select the places you selected? I mean, how do you make the decision to go this place but not that place?
So our initial sites that we went to were because we had been given reports from Eyes of the Reef. Eyes of the Reef program is something that it's part of the Rapid Response Contingency Plan through the state of Hawaii, and
It's a state program.
This is a it's a state-funded program, and what we have going on is we actually have the public reporting to us when they see instances of disease, and we got a lot of reports coming out of Anini, and so that's where we started.
Mm-hmm. Okay, when you go, you take the camera, you take something to take a little material with you.
We take a lot of material with us. When we get in the water, not only do you have a really thick wetsuit on because you're going to be in the water for a good 8 hours, we also bring transects, we bring sampling equipment, we bring things that we can look at.
When you float it on the surface near you.
No, we hang
You
it
have a
off of
boat.
our belts. We wear belts around our weight belts, and we just clip everything off onto us. So therefore, we're not leaving it anywhere on the substrate. We're not dragging it around. We're responsible for our own stuff, and we're making sure that we're not doing any damage as we're working.
Okay, what about your feet? I mean, what happened? I mean, when I step on a coral reef and I get infected, I get holes on my feet.
Oh, I don't step
get
on
infected.
coral ever.
Because you know that hurts the coral.
I'm
hurts
a-
them more than it hurts you, but it hurts you too.
I'm a
Yes.
floater. The second I get in the water,
Floater.
I put my belly straight down, and I just will float out to where I need to go to.
But
Sometimes
what
I
about
would,
scraping your belly on the coral? Doesn't that hurt?
I just I find the path of least resistance.
But you
and
know,
make
if
it through.
you're in the water, pardon me, but I really have to know about this. So you're in the water, right? Your belly is out there. You're flat. You're not stepping on anything because you don't want to do any damage, okay? And the wave comes, and then the wave deposits you ever so gently right on the coral.
Oh, that's another thing too. A lot of the sites that we go to, we are very cautious of where we're putting ourselves. We won't be surveying in breaks. We might have to get through a break to get to a survey site, but it won't be anything that's going to be shallow.
So you go down. You want 10 feet or so, and then you go down. So
Yeah,
you
we
never
just-
get deposited on the
Yeah,
coral.
no, yeah, we make sure that we're keeping ourselves and the corals as safe as possible.
Okay, so you
Safety's
get out of the
number one.
water, and you have this belt with all these bags on, and you go straight away, to see Blake. Blake is not there. Blake is back here in Oahu, in Manoa, right?
Yeah, we've had to ship
You-
things back to Blake
Yes.
before.
So how do you
Blake,
do that? How do you ship a little box? What, in the airlines? What?
Well, we have special containers. They put the samples in, fly it over to me, and I meet it at the airport and rush it back to campus.
And no, he said
What do you
campus.
tell the
We do
airlines?
this at campus. All of our work, our bacterial studies we do with this disease is done on campus, very far away from the water here on Oahu. And so,
Oh, because you don't want it leaking out into the water.
No, so
That would then you'd be making a mess.
yeah. Well, so all the work with the bacteria and our putative pathogens, we actually do it in an actual microbiology laboratory. So it's a Biosafety Level 2 laboratory. We take all the precautions. Always remember your gloves.
That's for your protection.
Yes, for
Biosafety 2 is pretty serious, isn't it?
it's more of a standard laboratory.
How does it work? Biosafety 1
It's
is
no threat to humans.
no threat to you. Bio 2 is a threat of some kind. Bio 3 is serious.
Yes, Biosafety 3 is usually the
Medical school.
Yeah, the very nasty things
Yeah, like
human diseases.
Antigens that could kill you. Yeah, like that. Yeah. Okay, sorry. So Bio 2 then.
And then, well, so we take all our precautions. We're able to run our experiments in a very controlled and contained environment. So when they bring me the samples, then I am there to try to identify what they have. So, for instance, on Kauai, we have that. You remember that black line on the disease lesions? We were able to isolate the Cyanobacterium that's part of this disease. So Cyanobacterium, if you
That's a cyano, C-Y-A-N-O
CYANO.
yes.
bacteria. Okay.
So it's a filamentous bacterium. So you don't have individual cells; you have them in a chain for this particular disease. And it's lucky for us, we can actually see this bacterium with our eyes. So if you,
Wow.
yeah,
Wow.
it's actually quite large. Each individual cell.
Do they move while you watch? Do they?
Oh yes, they do
They
actually
do
move.
They're motile.
Scary. And if you put your hand, you could touch them with your finger.
Well,
would they
I try not to
immediately-
touch the bacteria with my hands. I typically use a pipette or a forceps.
But if you did, what would happen to you? Would you be sitting here healthy today?
Well, we don't believe it infects humans at this point. Well,
We haven't
I mean,
seen any
you
cases
could
of
hold
human
one with
infection.
your hand. You could pick it up.
You could, but I mean that's something that we don't do. I mean, first off, like you said, we are a
No, of course.
microbiology lab.
Yeah.
We
It's a
use techniques
laboratory,
that
science here.
we're not going to do that. But
But it's big enough so that you could actually do that.
it's well, no, it's
for a bacterium, it's
for
very large.
yeah, it is large for a bacterium. I mean, you could see it. You can see the individual filaments, but it would be very hard to pick just one up because they are sort of attached in this mat to each other. You sort of have to tease them out. When we do grow them up on media, they do move really fast for bacteria that's
Okay.
of this size.
You're not cruel to them. You
Absolutely
don't like
not.
crush them like little acorns, like little, peppercorns.
No. We're there to study them. So
Study them. Okay, so there they are, and they're in your laboratory, and you're studying them. What do you do to study them?
So we look at a bunch of things. So well, first we try to isolate them. So it's when you get samples from the environment. Well, you can't just look at it and say, well, it's that particular cell. You actually have to tease them out and isolate them into different pure cultures and really see, like, okay, what is there? That's how we were able to find the Cyanobacterium, but it wasn't the only filamentous bacterium there. So we had to take some time, and we have several protocols how we isolate bacteria. And then we found the Cyanobacterium. Then we started looking at, next step is we have to grow it. Because if you can't grow the bacterium, it's very hard to study because
you want to grow it into a culture.
Yeah.
Then you can see what it does. You can
Yeah.
test it against it. But how do you know that the bacteria right there in front of you is the one that's creating the problem with the coral? I mean, it could be there at random, couldn't it?
Yes.
I mean, how did it get in Christina's pouch is the question.
So we do have we have Dr. Terry Work at USGS. He's a veterinarian, and he's done histology on these samples, and he's actually seen it looks like a filamentous bacteria and another bacteria that are penetrating through the cells that he's been able to see.
That means they attack the coral. He's been able to
see
them
that
attack the coral.
that they're there, that they're present within the tissue.
and doing damaging things. If they're
Yeah.
present there, it means they're sucking off the coral somehow.
They also could be creating an environment that they put out a lot of byproducts. The bacteria that we work with, they're not by themselves. There's a suite of bacteria that are normally associated with Blackband disease, and
Blackband is a coral disease.
yeah, that's what this disease is. And this is the first time that this has been reported at outbreak levels here in Hawaii. Up to this point in time, it hasn't been anywhere north of American Samoa at the levels that we're seeing it right now. But the bacteria that are there, they create an environment that is conducive to the sloughing of the tissue. It becomes a very toxic environment across that mat.
So there's no chance that these are symbiotic type bacteria. They're not there to help.
They're there to help each other, sort of, kind of. They like holding hands and making a little mess as they cruise along the coral.
Little toxic mess.
Little toxic messes, yeah.
So now you've got them all there and you cultured them. Then you want to find out what they do to the coral. So now you got to take the culture and put it next to or inside or nearby a coral and see what they do to the coral. Are you able to observe that?
Oh yes, one of the first experiments we did was a transmission experiment. So we had an infected fragment and we put it right next to a healthy fragment. We actually see this mat of filamentous bacteria actually move from the infected fragment to the healthy fragment and begin to infect it. Then
Chemically, what does infect mean?
infect. We're not entirely sure on what the actual process is, but it is actually moving onto the coral tissue, and then the tissue starts to die, and it's basically sucking up all the nutrients released by the dead coral cells. So
Why does this suggest to me that what you learn about watching these cyanobacteria attacking a coral cell is something that might be even more universal, and it might actually relate to an anthrax bacteria attacking a human cell or something, right? There must be a common denominator, but hold your reaction on that. We're going to take a short break. Okay, we can come back to it. Okay, that's Christina Runyon and Blake Ushijima listening to me and my ranting. my scientific ranting. This is Think Tech Talks on the SOWEST Hour. We're talking about saving our coral here in Hawaii. We'll be right back after this break.
I'm Hong Jiang, host for Asia in Review on Tuesdays.
And I'm David Day, host for Asian Review on Thursdays. Both of us broadcast our respective shows at 4 p.m.
And my topics tend to deal with questions related to environment, culture, history, and sometimes human rights.
And my shows tend to be on international business, foreign policy, geopolitics, and national security. And you can watch our shows live on the ThinkTech website at thinktechhawaii.com.
And you can also watch us on YouTube or Olalo.
So come join us on Thursdays at 4 p.m. I'm David Day.
And on Tuesdays at 4 p.m., I'm Hong Jiang.
Aloha.
Aloha.
Okay, Blake, we're talking about what you find down there and how this little infection process works. And you're trying to figure out why there's so much of it lately
yes.
and what exactly what metric are you looking for? What's your hype? I asked this question at the science fair all weekend. I went to the science fair. I'd always ask them, what's your hypothesis? Because everybody there is like a researcher in their own way. I love that when kids do that. I like it when adults do it too. So what's your hypothesis that you're working on?
Well, the first thing you have to remember is the main goal of any bacteria is to divide. And how do you divide? You need nutrients. So basically, the relationship of the coral to the bacterium is, well, the coral is food. But what we're trying to figure out is under what conditions does it allow this, ongoing war between the coral and the bacteria? What allows the bacteria to basically start winning and start basically eating your coral?
And multiplying in the process.
Yes.
They get stronger. It's a war, yeah.
Yes. So we're looking at several different things. First off is temperature. Temperature has been shown to play a huge effect on different coral diseases as well as many different other diseases, both for humans and animals.
Wild guess, the warmer the temperature, the more active the bacteria are.
A lot of the times, yes. A lot of the times, the hotter it is, the faster the
I'm
bacteria
going to
will
ask you
grow.
about climate change in a minute.
We're also looking at other things like different stressors. So freshwater input is that, of course, you have a marine animal, the coral. Is there freshwater input? Is that stressing the coral to a point of, well, now its defenses aren't able to keep the bacteria at bay? Or nutrient input. So is there runoff or fertilizer input into the ocean? So bacteria, main purpose is to divide. If you give it nutrients, they're able to divide faster.
Sure, they're off the plantations. That red mud runoff, yeah, has an effect on all this.
Well, we're still looking into this, but
Yeah, we're still trying to-
Okay, I've already made my conclusion. You can look into it if you want. Sorry.
We're just hypothesizing.
Okay.
Or if there's an animal vector, is there's something that's actually spreading this disease? Is it living on another creature that's motile, and is it going to each different coral colony spreading this disease, as like a bird would spread the bird flu or whatnot?
So there's a lot of possibilities
A lot
here.
of possibilities.
So
Yes.
where are you? I mean, have you narrowed it down, or are all of them relevant?
Well, we're basically just beginning. I think we only started pretty much last year, but we've made a lot of headway. We've identified what we think is the cause of this
So
disease.
your ultimate purpose here is to stop the onslaught. Yeah?
Yes,
Save the coral.
yes. To cure, you must understand. So we're trying to understand what these little critters are doing, what makes them tick.
So you might find that lime juice, for example, is spread over that area would save the coral. Who knows?
Fix it like the Koki frog?
So what about sea change? I mean, climate change, it makes the water warmer, right? The water is getting warmer, and that must have an effect on this.
Well, and not only that, you also have, sea surface, the level of it rising as well. So you have a lot of things that can be affecting it. And if you do have this change in your environment, are the organisms there ready for it? Have they evolved fast enough in order to be able to handle 1 to 2 degree temperature change? And another thing we need to think about is that if we have this warming effect happening, what is now the range of some of the diseases that were just within a small band, and now the temperature has changed and these diseases are able to move elsewhere or proliferate out? A lot of the things that we need to understand is that bacteria are ubiquitous, and that these
What does
bacteria,
that mean?
that means they're everywhere. They're
In the water,
everywhere.
they're in the water.
They're everywhere. They're
But
on
they
you,
only-
they're on me,
They only
they're
have
on
an
this
effect
table.
when the object, the victim, is vulnerable, right?
Yeah, when it's
Yeah.
susceptible. But now when you have this range expanding due to the range of the temperatures expanding, you can be allowing for more. You can be allowing for more things to be coming in. You can be allowing for even different fish species to be where they weren't before. So something that we need to think about is that the ecological changes that are going to occur, even from the bacterial level up to the larger fish level with global climate
I mean,
change.
that are
Oh, yeah,
occurring.
they are occurring.
Yeah. It's hard to save the environment. It's hard to do that. You guys, that's what you're doing here.
Yeah.
You're saving the natural environment. It's scary because you could fail. I'm sorry to say. Not true. After
Well,
all this.
That's part of science.
Sure it is.
You know,
It's part of
you
your
have
science.
you have
Yeah.
lots of failure before you have any success. You'll have 20 failed experiments before you have one that shows you the little inkling of hope that it's gonna work. You know, so the one thing we try to do is first observe what's going on, and then from there try to untangle the events and try to figure out what are the stressors. You know, what's the nearest pollutant source. You know, how many septic tanks are within that area. You know, we can do a lot. We're interdisciplinary. We don't just sort of stick to one thing. We use a lot of different tools of science in order to be able to look at it at all of the levels of what's going on with the disease.
And you've got to see it all. You've got to see
Absolutely.
all the factors that are out there, that could be out there. I mean, it's like sleuthing, isn't it?
Yes.
CSI. You know, that's why you guys are in it. It's the sleuth part.
Yep.
Also, that you like the animals.
Oh, I also like the water. I'm sort of privy to being more in the water than anywhere else. So.
Those great coral detectives.
Yes. Yes.
We'll take one last break here and come back and we'll talk about your challenges and how you overcome them in the program and in the funding and in the research. And we'll talk about the future, where this is all going to go and how it's going to dovetail with other things that are going on at SOWEST and elsewhere in the world. Okay, this is Think Tech Talks. We're here at the SOWEST Hour, saving out coral with Christina Runyon and Blake Ushijima, who are graduate researchers at UH-Manoa. We'll be right back after this break.
Aloha, I'm Nicole Horry for Think Tech. For nearly half a century, the Hawaii Foreign Trade Zone No. 9 has brought the benefits of the Foreign Trade Zone program to Hawaii businesses and entrepreneurs. DBET, the Department of Business, Economic Development, and Tourism, operates Hawaii's Foreign Trade Zone program to encourage international business and economic development. The Foreign Trade Zone's mission is to increase the amount of international trading activity in Hawaii, thereby providing employment opportunities for the residents of our island state. For more information, see ftz9.org. I'm Nicole Horry. Mahalo.
Okay, we're back. We're live. We're Think Tech Talks on the SOWEST Hour, saving out coral with Christina Runyon and Blake Ushijima, graduate researchers at UH-Manoa, the Hawaii Institute for Marine Biology, which is part of SOWEST, the School of Ocean and Earth Science and Technology, which is actually dedicated in many ways to saving our environment. So you have a team. You guys are not alone.
Nope.
No man or woman or combination is alone. So who's on your team? How
Well,
big is it?
From UH-Manoa, we have, in our lab, we have 5 grad students that have been instrumental in looking at what's going on. My field partner, Sylvia Berman, she's from Switzerland, and she's in the microbiology department, and she comes out with me into the field. And then we have Patrick Vidot, who's been instrumental in helping with the culturing, helping with the identification. And then we have Amanda Shore-Maggio, who went out on our initial trips and looked at what was going on, and also established some colonies for us to follow through time, as well as started a sort of a treatment. And then we do have
Lime juice?
and then we do have another lab member named Andy Berger, and he's just come on with us, and he's been instrumental in giving ideas and, you know, trying to get us to think sort of outside of the box on what could be going on, because he's
So everybody has a different role. They're not all just
Yep.
doing the same thing.
Yep, and so that's just within our lab. And so we're under Dr. Greta Abbey and Dr. Sean Callahan. Outside of our lab, we have Dr. Terry Work from
Okay,
USGS.
how many people you want to name? We only have a few minutes left.
Okay,
Yeah.
and I just have one more. We have NOAA. We have people from NOAA that come
Okay.
out. So we do have a lot of affiliations with USGS and NOAA as well.
Okay. How about you?
Oh, well, I'm just part of us.
Oh, you're part of the same team?
Yeah, I'm
Yeah.
just a
Okay,
small
all right.
member in a big team.
Yeah.
Okay,
We're
but do you
Team
have a lot of
Coral.
people in the laboratory?
I've had a few undergraduate interns. My most recent one, Donna Pascablo, she was part of the MARC program, the Minority Access and Research Opportunities, and it's more of an interaction between all the field guys. So they're
You meet on a regular basis?
yes
Every day.
Yeah.
Good. Where do you meet? At the school?
In the
school,
lab.
or
Okay.
if they're on the field, I get emails or calls.
It strikes me though that Hawaii, with what we have in a way of coral and all these factors and vectors you talked about, has to be unique in this area. It has to be one of the best places in the world to do this kind of. Am I right?
Oh, absolutely.
Are we doing world-class research? here?
We are doing, I believe we're doing world-class research. I mean, like I said, we're doing interdisciplinary work. We're not just focusing on one little aspect. We're bringing in as many different disciplines as we can to try to figure this out. And I believe it's a great laboratory for us to start looking at,
Where else are they doing it? They do this at Woods Hole?
I believe so. I noticed,
Yes, with
yes,
school.
and also I know they were looking at what was going on in the Florida Keys before the Florida Keys crapped out. I mean,
The end of that.
yeah, I mean, but there still are pockets. There's still places.
Okay.
There's still a little glimmer of stuff. But I mean, there's been. You also have in Australia a lot of work that's been done. And like I said, we've been to American Samoa, and there's work being done there as well. So there's a lot of places.
What's interesting though is Hawaii, we have healthy coral in some places, and we have diseased coral in other places, and we have all these factors and vectors and whatnot. And this is a place where you can see them both, you can see the process, you can try to figure out what's killing the coral. If you went to a place where all the coral was healthy, you couldn't do that, right? And if you went to a place where there was no coral or it was all dead, you couldn't do that. So we're in the middle. That's what makes us unique. No?
Yeah.
So what are the challenges to your work? Where do you get stuck?
Well, as a lot of scientists will tell you, well, you can get stuck pretty much anywhere.
Oh, nice.
But recently, the main problem has been the funding. So, funding's been tight from everybody in the scientific field.
But where has it come from, traditionally? What are the sources usually?
Traditionally, it's from the Department of Energy, the National Science Foundation. We have had funding from NOAA. We have had grants with NOAA.
We've had from TNC, from to go to Palmyra.
Oh, yes,
we've
Major
had
Conservancy.
from the state as well, DAR, DLNR, and like I said, SOAS has helped us. Sea Grant, we do have capability of getting funding, but funding is really, really tight. Both Blake and I, we're not just researchers. I'm a teacher at the university. And,
You teach also.
and-
You teach also. Blake?
Yep.
Yep. So we both have to, in order to pay for our tuition, this is what we have to do. So we get a salary as well and tuition waiver, but we don't. We're not just the lab people.
You're the troops. You're the troops there at UH-Manoa. You're the ones who make it happen.
Well, we're trying.
The working stiffs, as it were.
Yeah, absolutely. We are the working
Working
stiffs.
stiffs.
That's correct. That is correct. I mean, I still am in the process of taking classes and stuff, so it's not like it's just.
You're
all-
taking courses.
Yeah, I still take courses.
Yeah. You got both of you guys in a program for I don't know what you have.
I'm in the
now.
marine bio program.
That's a master's or PhD?
PhD.
PhD. And how about
And a
you?
PhD for microbiology.
Yeah, so you never stop for a minute. In fact, all of this that you're doing is sort of en route to that same direction. Isn't
Yes.
it? So, yeah. So what happens when you get the PhD? You take off and go somewhere else, or do you?
Go do a postdoc somewhere else.
Yeah.
Hopefully find a nice job someplace warm and sunny.
Yeah.
How long does it take? People say it takes a long time to get a PhD at UH-Manoa. You can speak freely.
I'd say
Nobody's listening.
I'd say the average is around 5 years.
Is that too
5,
long
6
or too
years.
short, or what?
No, actually, it depends on what you're doing. So
Mhm.
if you're working on something,
Very complicated. It's going to
Yeah.
take you a very long time. Like
or something that grows very slowly.
exactly. So like I said, this is my third year in, and I probably have about 3.5 years left, maybe 4. So I'll.
So it is a defined period of time. It's not, you can plan it.
I can try to plan it all I like, but it doesn't mean it's going to happen.
Life is like that.
Exactly. I know this. Yeah, I know this very well.
So if you had to look forward now on the program and your participation in the program, from all that you know, from all that you do all the time, you spend passionate days and probably nights working on this. Where is it going? Where is it going in the context of, its own development, its own sophistication, its own depth of science, and where is it going in terms of reporting to, what did you call it before, reporting to management?
Yeah. Well, I feel like this project in itself has just opened the door for coral disease research in Hawaii. It's one of the projects or one of the labs that's helped open the door. So hopefully more and more people will start getting interested. We have more graduate students, more professors getting interested, and just have people aware that this is a growing concern that we still don't know as much about as we should. So hopefully we'll get more research done and hopefully more funding, so we'll be able to do more research and try to, save our corals and save our reefs.
And then another thing that's really important is outreach and education. That's something that we do a whole lot of as well, and that's why we're involved in Eyes of the Reef program. So we'll actually go to communities and train them on how to look in their own ice box out there. And if they see something that just doesn't look right, how to report to us. And then we also have a second level of this where we will bring people out in the water and train them on how to assess what's going on, how to sample, and we go to schools. We try our best to get the message out that it's everybody's responsibility to really be taking care of their environment. And if you start with the keiki, it helps a lot, because then they talk to their parents, they talk to their aunties and uncles,
Yeah.
and you get more people out there. You get more people that are concerned and care.
You
about
take
what's going
a
on.
minute and talk to that camera 3.
Camera 3. Hi, camera 3.
Those are the people, okay?
Hi,
They're
people.
watching you now. Okay, what is your message to them about being part of the effort to save our coral and thus our environment?
Well, each of us on an individual level, we need to make choices on a daily basis of what we're going to do. Are you going to open that can of soda and drive that extra 2 miles? I mean, it sort of sounds like a hippy-dippy kind of way to think about it. But if you really think about the impact and the footprint that you're putting on the planet on a daily basis, you might start, taking a little bit of that footprint back and try to make a little bit of a difference.
Like?
Well, what you can do is, I'd say, is that you can actually help us. So Chris mentioned the Eyes of the Reef program, that where if you go to Reef Check Hawaii, you can go on their website and look, search for Eyes of the Reefs. It's a community-powered program, basically. It's so if you can go, you're in the field or in the water, you're just floating around, you just see a coral colony that just looks funny, you can report it. Or you see something that just doesn't seem right to you. I mean, this is everybody's ocean. This is everybody's island.
Yep.
And we're all responsible.
We have a Facebook page for that one.
What is it?
As well. Eyes of the Reef.
Okay, Eyes of the Reef. Look it up, Eyes of the
Eyes
Reef.
of the Reef. We
Google.
also have a blog called Reefology 101. It's all one word, and it tracks all of the work that we've been doing. We put it up monthly, and it's from what we do in the lab to what we do in the field to what we do with outreach and education. So you can follow us there.
Think Tech Talks, the SOWEST Hour, Saving Our Coral with Christina Runyon, graduate researcher, and Blake Ushijima, graduate researcher at UH-Manoa Department of Microbiology at SOWEST. Thank you very much, both.
Thank you.
Thank you.
Aloha.
Aloha.